And Jess Lindstrom, master's student in Jill Hamilton's lab, is going to be presenting work for part of her thesis. So welcome, Jess. Fake, quiet clapping.
Wait, can you see?
Yes.
Okay, you can see my presentation.
Yes.
Okay. All right, so thanks everyone for coming to my talk. I'll be presenting my graduate research entitled Seed Sourcing for Climate Resilient Grasslands and the Role of Genetic Variation During Restoration. So obviously we're going to be talking a lot about grasslands, but let's first define what a grassland is so we all get a pretty picture in our mind's eye. These are very nutrient-rich natural habitats that exist from up in Canada, across the US Great Plains, and into Mexico, and are typically dominated by grass and non-woody forb species. And of course, they're characteristically devoid of many trees. And then a fun fact is historically the Great Plains region, bison were the dominant grassland grazing species and acted as long distance seed spreaders. So jumping back in, we should care about grasslands because they're biodiverse, very nutrient rich habitats. They provide a myriad of essential services and functions, including carbon sequestration, nutrient cycling, maintaining soil health, water quality, and are essential habitats to a host of species. However, despite their importance, grasslands are in decline, primarily due to those nutrient-rich soils, which has led to a conversion of up to 99% of native prairie habitats lost to crop row agriculture. This has left landscape really fragmented and sparsely located across grasslands sparsely located across the landscape, which has reduced connectivity and may further degrade those remnant habitats, which may allow for increased rates of invasion by non-native species. So while these habitats may be declining, I do want to say that there is hope through restoration, with the priority being to recover those ecosystem services and restore connectivity across the landscape. So like I've mentioned, we know grasslands are essential habitats and they need to be conserved and restored when possible. Restoration is a huge industry, but it's not without its own unique set of challenges. And so with my project, I will advocate the need for restorations to have a two-pronged approach, one that includes using species diversity and additionally including considerations of within species genetic diversity. And by considering both aspects of diversity will be essential in ensuring that these habitats are able to persist over time. And so I mentioned species diversity. Traditional restorations are really doing a great job in considering species diversity. We know that a functional restoration has to have a variety of different plant species as they serve different roles within a habitat and may even provide a buffer against invasion by those non-native species that are competing for the same resources. However, the within species genetic diversity, meaning the raw material that natural selection is going to act upon, is not always considered. And the typical approach to doing a restoration is a local is best for getting our seed. So what this means is a local seed sourcing approach applies on the concept of local adaptation. And so in this figure, if you can see my mouse, the x-axis represents a climate gradient, and this line indicates a site that we're going to say we're doing a restoration. And so using local adaptation or local seed sourcing approach, we only want to collect seed from nearby our location that we're doing this restoration, assuming that this seed is already locally adapted to our environment and should show the highest fitness on the y-axis. However, this is an issue because with this seed sourcing approach, we don't typically know what the scale or extent of local adaptation is. And we also know that climate is not consistent through a species' entire range. So our curves here really represent different scales or extents of local adaptation, in which we may have very high selection, indicated by this more narrow curve, in which we only want to collect seed that's locally adapted to our smaller climate range relative to where we're doing a restoration. But again, we don't know the scale, and we could have more broad levels of selection. I don't know if you can see my mouse, more broad levels of selection in which we could actually move away or further away from that site to collect seed from a greater distance and still maintain high levels of fitness in that site. And additionally, we also know that climates are changing. So how will these changing environments affect our expectations of local adaptation, especially when we often use historical references to guide restoration? So in this figure, we have an original climate at a known location where we're going to do restoration. Now climate has shifted to the right, and we may not know how that's going to affect local adaptation, especially if we only collect local seed. So given climate change, will locally adapted seed be able to adapt at a similar pace to adapt to these new climatic conditions? These are some of the questions that will impact how we decide to source seed for restorations, and potentially moving away from a local is best strategy to potentially including more unique seed sources for successful restoration. And so, like I said, our often advocated strategy is a local seed source relative to where restoration is occurring, but there are more seed strategies that are being advocated for and still need field testing. So in this graph, we're just demonstrating some of the different sources of seed collection strategies in which we talked about local. So we have on our x-axis the maintenance of local adaptation. And if we have a seed restoration if we have a restoration we're doing at the star, so that's wherever a restoration is occurring. My mouse keeps disappearing, sorry. And we only want to use local seed for that, maintaining local adaptation. We're only going to collect seed very close to that star, assuming that this will show the highest amount of fitness and survival for our restoration. Another advocated approach is a genetic variability admixture approach, which aims to increase the genetic variability. And so you can see we have a star representing, again, that restoration on a landscape. And instead of only collecting local seed, we have a variety of different populations across the climate gradient that we're going to collect from. And so what I'm going to do in my project is to test a more regional admixture approach, which aims to kind of hedge our bets by both trying to maintain local adaptation on our x-axis and include considerations of increasing seed sources from within a region to maintain genetic variability. So this brings me to my 2 project goals. One, which is, does genetic diversity matter within a restoration? And to do this, I'll be using a single source versus a multiple source seed mix approach in restoration plots in both Minnesota and in South Dakota to evaluate how plant community composition changes over time. I'll also be doing a second part of my project, which is looking at, can a species climatic niche inform restoration? And to do this, I'll be looking at a species presence across the landscape, its known climate at those locations, and then using that relationship to predict habitat suitability in the present time and looking at how that might change under climate change scenarios. So we'll start with my genetic diversity component with my restoration small plots. But before we start, we first need to know where on the landscape our native species occur. And this is a particular challenge within the Dakotas in just knowing where do we find seed to source, whether we want to source it regionally or locally. And so in the summer of 2019, we had 6 different field crews across 6 different regions collecting seed in Minnesota, North Dakota, and South Dakota. We mapped 26 different species across native remnant prairie sites, and we use the Esri ArcGIS collector application to do this mapping. And this is a great application because it can be used on tablets or mobile devices for real-time in the field data collection. So here is a pretty slide that shows the 26 different species we mapped and later collected seed from. We chose these species because not only do they flower and set seed throughout a growing season, they also represent common prairie species with wide distributions that are commonly used in seed mixes. And so we have on the top here some of our early flowering forbs. We have our asters, some legumes, and then also some warm and cool season grasses as well. So now that I have these populations mapped, I want to bring us back to my goal, which is to use different seed mixes on experimental restoration plots in Minnesota and South Dakota to look at that community composition over time. So like I said, we first mapped species and then we had to go back various times throughout the season to see if seed was viable and ready for harvesting. And so we would return to a site on our collector app. It would look just like this top photo here, in which each point represents a species we mapped. We followed a strict protocol in which no more than 30% of a species was harvested from a site. And then we also use seed bags labeled with the location of origin we collected that species, as well as the species code. So once we had seed, we would then bring it back to our field site to prevent any molding and maintain seed viability. Seed was then weighted to know about approximately how much we collected, and then it was cold stored for future use. All right, so my first semester and most of the second semester at NDSU was really dedicated to cleaning all that seed we collected. And I really have to thank all the undergraduates and the graduate helpers I had, and also Jared Pacifica at the USDA, who also let me use some of his equipment. And so the majority of our seed cleaning was hand stripping seed from stems. And for some of the more difficult species, like on this top left picture, we ran seed heads through a seed cleaner, and we would end up with clean seed ready for use to make seed mixes. So in designing my seed mixes, again, I want to look at a single source mix versus multiple source mixes. And so species had to be collected from a minimum of 5 different locations. So I could have 5 single source mixes versus a multiple source, which includes seed from all of those 5. And from the 26 species, we retained 14 of those for use in our seed mixes. This is partially just due to the fact that we had 1 season to collect all this seed, and so we just kind of made do with what we had enough of. And so for that, I used the lowest available seed weight for species to produce my seed mixes, as that was kind of my lowest common denominator. And then seed mixes were also based on a latitude to account for some phenological differences. So here is a proportional seed map, which I'm using to show how I made my mixes. And so we can see that each region is color coded. So my blue tones represent my South Dakota seed mixes. My red yellow tones represent my Minnesota based mixes. And so we can see that the dark blue color here indicates my first seed mix, so seed mix A, and it includes approximately 8 species from 1 single location. We can see I have near that latitude a couple smaller dots of dark blue indicating other populations in which I was able to collect species from for that mix. So the main point of note here is that there's a mild color gradient from north to south, indicating the different latitudes in which I made my seed mixes. So now in designing those seed mixes, it was a long process. So I'm going to try to concisely explain how I did this using this photo. And so we will say that each of these bags at the top represents one unique species, in this case, lead plant. And so each bag represents a unique population. And remember, I had to have at least a minimum of 5 locations for my 5 source plot. And so we have 5 different bags. I used my lowest seed weight as my low common denominator for the maximum amount of seed I could use. And so in this case, we'll say the lowest amount of seed I had for lead plant was this black population. I split that amount equally because I have single source plots and I have a multiple source plot. And so my single source plot, I have 3 replicates, and then I have my 5 source plots, which contain the same proportional amount of seed. So I divided my lowest amount by 4, and then I had 3 replicates of a single source mix. So from that, I had 1 replicate, 2 replicate, 3, all of the same seed mix of that one unique population. I then took my leftover amount of that 1 source and divided that by 5, because my multiple source plot includes all 5 species, all 5 unique populations of that 1 species. So in total, it's the same proportional rate. It's just the 5 source mix includes the 5 unique species for that 1 population, or for 5 unique populations for that 1 species. So once all the seed mixes were produced, it took a super long time. It was a lot of fun. We ended up with a total seed mix rate of about 57 pounds per acre in each seed mix, which is about 5 times the normal recommended amount in restoration. But in our case, we had very small plots and we were okay with this because we wanted to give our restoration plots the best chance of establishment to monitor over time. So as a reminder, we have plot sites in both Minnesota and South Dakota, and we set up plots to evaluate single source mixes and multiple source mixes to look at community establishment. So in 2019, we also prepped our experimental plots in our 2 study regions. The first one is again in Minnesota at the Regional Science Center. And this is a site owned by the Minnesota State University Moorhead. This acts as a research center, and until 2014, certain parts of the center operated as a golf course and are now being restored to native grassland habitat. Our second site is in Leola, South Dakota at the S.H. Ordway Prairie Preserve owned by the Nature Conservancy. This site operates primarily as a hayfield and is a pretty homogeneous community at this site. For designing my experimental plots actually on the sites, I followed the same protocol in both regions, starting with my 5 single source plots. So these plots are only going to contain a single source for each unique population, and these are labeled as A, B, C, D, and E, and they're each replicated 3 times. I have a 6th plot I'm calling, for sake of brevity, A, B, C, D, E. It has 5 replicates and contains the same proportional seed from all 5 single sources. The idea being, again, that increasing the number of unique seed sources is a way to increase the within species genetic diversity, which may increase establishment over time. Some other things are each replicate was about 3 meters by 3 meters, and each replicate was maintained. We had a 3-meter buffer zone maintained, and each plot mix was separated by at least 100 meters to limit gene flow. So at our different sites, we had different setups. At the Minnesota plots, we really had to work around the site to get where we could set up our plots and stake everything out. We used landscape cloth as a light deprivation tool to remove vegetation, and then as well as we maintained those mode buffer zones. In Ordway, South Dakota, our site, like I said, was a hayfield, pretty homogeneous landscape. We use Roundup and mode to kill off the vegetation within the plots. So we have seed mixes, we have our sites set up. In April of 2020, we went to our plots to prepare them from seeding. And so these photos are from the Minnesota sites. And we can see that, so we took off that landscape tarp, and at this top photo here, you could see all that dead vegetation. So we had to remove that and then expose that soil to get ready for seeding. And I want to do another quick thank you to everyone who helped out during that day. But once our plots were fully prepped, they were hand broadcast seeded, and I used a push roller to compact the seed into the ground. The roller and my shoes were, of course, cleaned in between each plot to remove any risk of contamination. And then I also gave these plots a one-time watering to aid in those initial stages of germination. Throughout the growing season, we returned once a month to record community data such as species present, number of individuals per species, the % species coverage, the % bare ground, and the % thatch cover. And I want to give a quick thank you to Rebecca Hedlund out at Ordway, who helped monitor the plots out in South Dakota because I was unable to travel due to COVID restrictions at the time. So going back to the question, does diversity matter? We're using these 5 source seed mixes as a way to increase genetic diversity within restoration plots. With this, I predict that the 5 source plots will have an increase in establishment success when compared to those 1 source plots. And I'm really defining establishment success using common traditional restoration metrics, such as species richness, species evenness, and species abundance. So what did we find? In year one of our evaluations, I have just looking at my richness, looking at basically how many different species established within the first growing season. I have my South Dakota data here for looking at species richness separated by a single source mix A compared to a multiple source mix A, B, C, D, E. And I'm just using A for the sake of brevity because all single source plots primarily were reacted the same way. And then I have the month that we collected data observations, and I have it split up by the total richness, so the total number of species that established within a plot versus the planted species. So what species established that we planted? And as you can see, it's primarily weeds. We did have stuff established, but it wasn't what we planted. And spoiler alert, we saw similar trends in our Minnesota plots as well. So in our first year of looking at community establishment, we found a lot of non-planted weedy species that established, which makes sense in the restoration literature. We then had to modify our question a little bit to look at plot level community diversity. And I did this to see if there were any patterns in what our baseline community establishment looked like. And I used a Permanova approach to evaluate any significant differences in my community composition. So here is basically my South Dakota Ordway community diversity. So I'm interested in evaluating any differences in my community diversity, and I use plot group or seed treatment, my replicate, and my month as my main explanatory components in explaining community diversity. And I found that in South Dakota, month was my only significant to explaining community composition differences. So from here, I used a non-parametric multidimensional scaling approach, or NMDS, to actually visualize those differences in community composition over time. And so what we can basically see here is groups that are more similar in their composition are clumped closer together in 2D space, and groups that are more different are further apart. And my ellipses here show my 95% confidence interval in explaining or identifying similar communities. Again, what I found through my Permanova was that my community composition was mostly explained by month. And we can see that I have seed treatment as my colors. So the different seed mixes are colors, and the month was observed with symbols. And we can see a really pretty stark clustering of my symbols. So all of my June plots are clustered, and they're all contained within the ellipses. So these are all really one distinct community as compared to the other months. Where we do have areas of overlap just indicates that these communities may be a little bit more similar in their composition. But the point here is that we saw that the community was really driven by month and not necessarily the seed treatment we planted. I did the same Permanova analysis with my Minnesota data, and I found that my community composition was significantly different based on the seed treatment, as well as the replicate and the month, as well as that interaction of time and space. So how the treatments compared over months. And so in my NMDS, we can see that my colors are more closely clustered together, meaning that my site or my plot groups or my seed treatments are more distinct communities. And so what this is basically saying is where we put the seed on the ground mattered significantly in shaping community diversity. And I'm saying that primarily because we saw, as I showed with my richness, that not a lot of our planted species emerged. So I'm not taking this strictly as my seed treatment, but more as where we planted the seeds on the ground in Minnesota. And this really actually made sense because we can see some site level differences where we establish at the Science Center. Plot A represents a welcome center. It's very little disturbance. Plot B and C are by Dr. Hamilton's Prairie Smoke Garden and are along the road. We run along across the road by the railroad tracks. We have plots D and E, which were that area that operated as a golf course. And a little bit further, we had plots A, B, C, D, E, my multiple source mix, which is in that tree marshy area of the golf course. And so again, we can see just through this quick image that we had a lot of site level differences that we had to work with in making these seed mixes or making my restoration plots. And so location really mattered in establishing those communities. So now that we covered my initial community compositions in our plot, we found that there were site level differences, but that brings me back to my main goal, which was to look at how different seed source mixes influence community establishment from a single source approach to a multiple source approach. So we kind of had to modify our question a little bit. So here, did the same Permanova approach. In this case, I lumped all of my single mixed data together to compare that versus my multiple source community data and seeing how that impacts my community composition. And my prediction here was again that my 5 source plots should have an increase in establishment success compared to my single source mixes. So going back to my question of does unique seed sources per species impact emergence? Like I said, I use that Permanova approach, and I did find that they were significantly different, both by the Regional Science Center and in Ordway. And so my mixes single versus multiple were significantly different. But again, the caveat to this is that this is more related to the site than the actual seed treatment itself. And I included a visual representation of kind of what this looked like and how we can see community composition differences with some photos taken at Ordway, in which we have a single source plot that was a photo taken at July of 2020 compared to a multiple source plot that was taken at the same exact date and time. And so we can see just visually that we do have some community composition differences between single and multiple source plots. So my basic takeaway from this work was that restoration plots sown with the same species at the same time in different mixes had similar yet different patterns of emergence within the first year. I'm saying that they're similar in that both community in both regions were characterized by primarily woody species, with a small amount of our planted species establishing in the first year. And then for both regional communities, site history seems to play a huge impact in shaping those communities. However, we had different effects. In Ordway, where the site history is a much more homogeneous community, our community diversity seemed to be primarily influenced by month or the growing season. At the Regional Science Center, though, where we had a lot of site level differences, just within one area, community composition was primarily dominated by the location in which we planted, as well as the effect of time and space as well. So next, I want to shift into my second chapter, which is ongoing work in looking at can a species climatic niche inform restoration. And so to do this, I'm going to use known presence point location, indicating where a species exists on its landscape, the known climate at those locations, to then look at habitat suitability in present time, and then how that suitability might change given climate change. And so my first predictions here are that first, different climate variables may influence different species. And then secondly, that climate change may also affect those species range distributions. So why does this matter? Well, this is going back to that discussion of local adaptation and climate change. We don't know how communities will be impacted by climate change, and if they are impacted, will they have that capacity to adapt to a change in climate fast enough? Using these models, we can then make predictions of where habitats may be consistently suitable for a species versus areas that may become inhospitable, which may inform how and where we restore, and identifying areas of connectivity between these fragmented landscapes. So as I mentioned earlier, we have these ground truth presence points where we collected locations in Minnesota, South Dakota, and North Dakota. These are known presence points on the landscape. But because we have a bias in only looking at the range within our local area, I also combined observance records with the Global Biodiversity Information Facility, also known as GBIF, which hosts observance records as well as long-term herbarium data as well. So in explaining my modeling approach, I'm really going to use Dalea purpurea, or purple prairie clover, as my example species. Here I have my mapped collected points combined with my GBIF records to really show that species presence across the landscape. At each one of those points, I have a known latitude and longitude for those presences, in which I can pull out the approximate climate at those conditions. For modeling with climate, I will be using the Bioclim variables from the World Clim database, and these primarily relate to temperature and precipitation at an annual scale and also quarterly. Again, I'm going to extract the climate at those known presence points, along with some background data to then extrapolate to predict habitat suitability. Ultimately, I'm going to take a species level approach, assuming that not all species are equally influenced by the same environmental conditions. And so I want to find the most appropriate set of variables to reduce collinearity between some highly related variables, as this can reduce my model accuracy. So in finding my variables that matter for predicting habitat suitability, I have here my species again, DALPUR on the x-axis, and the percent contribution to the model that all 19 Bioclim variables had in producing my models. I then used a 90% cutoff as my limit for choosing variables. So here I'm showing that for my Dalea purpurea, the only Bioclim variables that really mattered in producing an accurate species distribution modeling were my mean annual temp, my diurnal range, which is basically looking at the mean of the monthly max versus the monthly min. I had my temperature sorry, my mouse keeps going away. My temperature seasonality, the max temperature of the warmest month, the minimum temperature of the coldest month, the mean temperature of the driest quarter, and the range of precipitation, and then the, there's my mouse. And then this one here, my top one, is the precipitation in the warmest quarter. And so I retained all 8 of these Bioclim variables out of the 19 original. And so even though this 90% line crosses the middle of this Bioclim variable, it's still counted in my variable importance. So again, I did this across all 26 species that we had collected data on. And the big takeaway here is that you can see a lot of different colors, which basically is showing that not all species were influenced by the same climate variables. So I have my species on the x-axis here. And again, that same percent contribution to making my models with that 90% cutoff rate. And I did find, though, that there were some variables that were more important in explaining more species variability. And these ones were the mean annual temperature, the mean temperature of the driest quarter, and then the precipitation of seasonality, which is basically just the annual range of precipitation. So now that I have those known presence points, I have known background points, the associated climates of the variables that are most important, I was then able to produce a contemporary climatic niche for my species. In this case, again, Dalea purpurea. And here we can see the red values indicate high habitat suitability, with the lower blue tones representing areas of low habitat suitability. Now, once I have our contemporary models, I wanted to then compare, will these ranges change given climate change scenarios? So in this GIF, I'm using a 2040 moderate climate change scenario. So we're just predicting in the near future here. And we can even see that, again, areas of red indicate very high habitat suitability, with the lower yellow to blue tones indicating low habitat suitability. And we can clearly see that, especially in some of our edge regions and that area of really high habitat suitability, we're decreasing in habitat. And we're gaining some in the north as well a little bit. But the main point here is that, yes, these ranges will change with climate change, even in just a moderate scenario. Here I have another species, Amorpha canescens, or that lead plant. And again, we can see that we had different climate variables explaining our climatic niche, but we still have some levels of range change. So not all species are responding the same to climate change. This one always takes a little bit to pop up, but I just want to point out again that I'll be doing this for all 26 target species we have data collected on from 2019. So this is an ongoing part of my project. I need to finish modeling my future climate suitability for each species, and then I will begin to quantify those differences in current habitat suitability versus those predicted climate scenario suitabilities. I will also be evaluating my models in comparison with other types of modeling approaches. As I briefly mentioned, I'm using a MaxEnt approach, which utilizes presence-only data, meaning that I have those known presences. I'm using those background points to predict habitat suitability, but I really want to compare how this prediction compares with other models, as each different model has its own sources of uncertainty. And this is really well demonstrated with this Grimmett et al. Paper that recently came out that looked at MaxEnt, general linearized models, and random forest models, and looked at the predictive accuracy of all three of these models. And so they looked at the area under the curve, which is a really common used method of predicting your model's ability to predict habitat suitability better than random. And so higher areas under the curve indicate that your model is able to discern those relationships between a species presence and a landscape to accurately predict suitability. And so each of these models, again, has its own sources of uncertainty. A general linearized model is pretty good at modeling compared to MaxEnt. The difference here is that it really uses, it traditionally uses a true absence data, which we don't have, but it can work with pseudo absences like MaxEnt. And so we predict that between MaxEnt to the general linearized model, there should be a similar output. And then with the random forest model, this one is a decision tree based algorithm, which uses these decision trees to produce areas of high accuracy for predicting models. And this one is really good at reducing overfitting. So saying that there are relationships when there's not, but it really only works well if you have a large data set and begins to overpredict if you have a smaller data set. And so I really want to compare how my MaxEnt models compare against those general linearized models and the random forest models. So for this section, in conclusion, we wanted to use a species climatic niche to see if we can use that to inform restoration decisions. We did find that not all species are equally influenced by the same climatic variables. And so far, I'm able to see that not all species have similar patterns in rain change under climate change scenarios. By understanding how each individual species ranges change, maybe where they are first off, so where each individual species range is currently, and then by seeing how those ranges may be impacted by climate change, we can then begin to make some community-wide decisions. So we could begin to make seed transfer guidelines on how we're moving seed across the landscape. We might identify potential areas of concern or areas of restoration interest, as well as areas of habitat connectivity across the landscape. So in summary, does diversity matter? At this stage of the restoration, our plots were dominated by weeds, so I won't give you an answer to that, but we did see that site history matters. And so it's important that we consider site history whenever we're going into a restoration, because the site history may have a significant role in impacting your first year communities. And this really demonstrates the need for having long-term monitoring of our plots. Secondly, are climate variables that impact communities the same across grassland communities? In short, no. These are not a one-size-fits-all. We saw that different species were impacted by different climate variables. And similarly, will climate change affect a species' ecological niche? Yes, but each species may respond in different ways. In short, different species do different things at different times. So I'm just going to go to this slide as we're wrapping up here. Again, I wanted to advocate for this two-pronged approach that for successful restorations, genetic diversity needs to be considered along with restoration efforts. It needs to be considered in restoration efforts along with that species diversity component. Restorations are not a one and done sort of project. They need long-term monitoring and maintenance if necessary. But by taking into account both of these aspects of diversity will be essential in assuring that they're resilient and have that capacity to adapt to change. With this, I just want to acknowledge all my funding sources, ECS and DSU, as well as my advisors, Dr. Hamilton and Dr. Allering, as well as Dr. Norland and Dr. Travers, my committee members, the Hamilton Lab, everyone who helped out in data collection, the different services that allowed me to collect on their lands, as well as everyone who helped with mapping and landscape crews and everything, as well as Jeff and Wendy, as well as my partner Evan and our pets for emotional support. With that, I will take any questions.
Nice talk, Jess. Very nice.
Thank you.
If you have you can either unmute yourself or put your question in the chat. So
the prairie dogs notice immediately.
There's a question in the chat. It says, did you calculate a diversity index for comparison of sites or evaluation of fitness / survival?
And how I use, oh, am I still there?
yes.
Okay, sorry, I just got right at the end again, I got your internet connection is unstable, but yes, so I wasn't maybe clear enough, but when I looked at community composition, I took into account, so I had species, richness, evenness, and abundance, but I ended up using Shannon's diversity index to look at community composition.
So
Jessica, oh, go ahead.
I was going to ask if you, for the restoration experiment, if you kind of dive down and look at species by species, are there certain ones that seem to establish over others? And if you did the analysis just within certain species, do you think you might be able to better test if for those species your seed mixture had an effect?
Yeah, I think really we're going to need longer term monitoring of these plots, because right now I had a lot of witchgrass, and so if I only looked at some of these species, if I just looked at what established, I only would have maybe 1 or 2 species and only a couple individuals, and so I don't think diving into just what we planted yet would yield any good results, because we really didn't have much, if that makes sense, if I answered that question.
Yeah. So do you think there's with the weed effect, is it seed bank, or are these weedy species migrating in? And it seems like that could have some implications for the plot size, would there be edge effects if you were restoring acres and acres where there'd be weeds around the edge, but maybe the center could be restored more effectively? Do you have any thoughts on that?
Sorry, I got lost in the question, but
yeah,
we did see, so yeah, there was, I think there was an effective seed bank at the site, and that's something eventually we're gonna try to look into, is because this is the at least at the Regional Science Center, where there was that golf course, there is data on what's nutrients are even available in the soil seed bank, and then as well as, I think it was just a lot of weeds that were already in the seed bank. And then I think in South Dakota, a lot of the species that established that were non-woody species or non-planted species were already kind of species that were really common out there, the absinthe wormwood.
Thanks, Jess. I think Donna had a question.
Yeah, I just wanted to tell Jess hi. I'm not showing my face because my hair is a wreck. I'm Jess's undergrad advisor, and she did turtle research with me and small mammal research, and she got her love of prairie going. And Jess, you did a wonderful job. You made me proud, and you sound like you already have your PhD. Although this is a master's, right?
Yeah, we're not there yet.
So far. Anyway, congratulations. You did a wonderful job, and it's good to see you. All righty,
Thank you.
bye-bye.
I see another comment. Do you want me to read it, or do you want?
Yeah, yeah, go ahead.
Okay, yeah. Mallory asked if this was the first time my target species had been used in distribution models. And I haven't looked at each 26 different individual species to see if they've been used in species distribution modeling. But I do know there's a really great paper that came out recently that was Lyon et al. And they used a more like, I'm going to say a shotgun approach, in which they used a lot of those same target species because they're very commonly used in restorations. And so they combined all of that species data and used the same climate variables to produce then like community composite level species distribution modeling, which I think is great. And I did use it a lot in referencing. But what they did again is they used all of those species and used the same climate variables, which I think is maybe a cause of concern because as my at least base data showed is that not all species are influenced by the same climate. They may be on the landscape together, but there is some nuance that we may be missing by just using them as one big community. I hope that answered the question.
Are there any more questions for Jess? All right, thanks, Jess. Really nice talk. You did it. Congratulations.
I wore my good shirt and everything.
Sorry, I don't want to burst your bubble. There's another question in the chat.
Oh, yeah. Sorry, I didn't see that. Oh, do you have any idea why seasonal progression was more important in South Dakota site, well, in Minnesota? And yeah, I think that's primarily the site history. So at South Dakota, it was like a very homogeneous community, just used for hayfield. And so I think it was more of the plots acting like the growing season for the homogeneous community was more important because everything is pretty much the same. And then whereas like the Minnesota site where we had those huge site level differences and we even had issues in where we were going to put on the ground, like spatially, I think that's why the site location was the most important part. So like it says in the model plot treatment, but again, we didn't have anything established, we had very little established of our species. So I'm taking plot treatment just as site because where we plotted, where we put that seed treatment was really site specific or location specific.
So I've also got a question.
Yeah.
So another issue involves succession. And a lot of your weed species are probably pioneering species, and you basically took your plots back to start in the succession. So what are your long-term plans?
Well, my long-term plans are to get a master's degree in skedaddle, but there are going to be long-term plans to monitor these in the future because just like any restoration work, there's usually continuous monitoring. And so I'll be monitoring them again this summer a little bit. And then I think there's talks with TNC and stuff to keep it going.
Okay,
I put in all this work. We're not letting these fail.
Thanks.
Yes, thank you.
I don't see any more. I think they're just, yeah, I think you've answered most of the questions. So nice job, Jess. Congratulations. Now you can do your victory dance.
Yeah, I mean, mostly I still have to do some work, but victory dance for today.
Yeah. Well, maybe at the defense, you can do the interpretive dance for your species distribution model findings.
Yeah, over lots of tears.